This video will look at gene expression, the process of taking the genetic code and turning it into a functional protein. We will cover chapters 5.6 and 5.7. Gene expression is a two-step process. The first step is transcription. This is where the DNA is read and used to make a messenger RNA molecule. The second step is translation, when we use that mRNA molecule to direct the production of proteins. In eukaryotic cells, transcription occurs in the nucleus where the DNA is housed, and the translation process occurs out in the cytoplasm. The product of transcription is an RNA molecule. Remember from Chapter 2 that RNA is slightly different in structure than DNA. For example, RNA is only single-stranded, not double-stranded like DNA. RNA has a ribose sugar instead of a deoxyribose sugar in that sugar phosphate backbone. And instead of thymine, RNA will use uracil as a base. During transcription, copies of the DNA message will be made. However, that message will be changed slightly as it is turned into an RNA molecule. We're still going to use complementary base pairing. When we transcribe DNA to RNA, we're going to see slightly different pairs than DNA to DNA. So, for example, if a DNA molecule has an adenine, it will actually pair with a uracil in RNA. A thymine in DNA will pair with an adenine in RNA. A cytosine in DNA will pair with a guanine in RNA, and a guanine in DNA will pair with a cytosine in RNA. The big difference is that first pairing, going from an adenine to a uracil. Let's practice this for a moment. Here is a DNA strand. Transcribe this information to an mRNA using complementary base pairing. Pause the video for a moment and write this down. Remember, thymine will pair with adenine, adenine with uracil, guanine with cytosine, and cytosine with guanine. So, our mRNA will read AU, AA, UC, AU, CC, AG. Our key player during transcription is an RNA polymerase enzyme. This enzyme is going to be responsible for actually catalyzing the reaction and creating the mRNA molecule. This transcription is a four-step process. First, our RNA polymerase will recognize a specific site in the DNA and bind to it. Second, it will transcribe the actual DNA, making an mRNA molecule. Third, it will terminate transcription. And last, that mRNA molecule will go through capping and editing. Step 1. Recognize and bind. RNA polymerase must first find the promoter site on our DNA. This site is very specific and allows the RNA polymerase to bind to the DNA. Once the DNA polymerase has made a connection, the DNA will unwind slightly, creating a transcription bubble. This allows the RNA polymerase to actually do its job. The second step of transcription is actually transcribing and making our mRNA molecule. RNA polymerase will pull the DNA through itself. As that occurs, the DNA will continue to unwind ahead of the RNA polymerase. Behind the RNA polymerase, the DNA rewinds back into that double helix. If we look at the transcription bubble, the RNA polymerase will be reading the parent DNA and creating our mRNA transcript as it moves along. We can take a closer look inside of the mRNA molecule. The blue strand is our DNA, the yellow strand is our mRNA. Remember, This process requires complementary base pairing. The DNA will be read, and a complementary piece of mRNA will be matched to each DNA nucleotide. The third step of transcription is termination. In this step, the RNA polymerase reaches a termination sequence in the DNA. This is just a section of DNA that tells the RNA polymerase that it's time to stop. The termination site is at the end of a gene. When the RNA polymerase hits the termination site, it falls off of the DNA and is able to go back and read another gene at a later time. The mRNA molecule is released and is now floating in the nucleus of the cell. The last step of transcription involves capping and editing of the mRNA molecule. At one end of the mRNA molecule, a cap will be added. At the other end, a tail. These ends are designed to help protect the mRNA from damage. The cap also helps the ribosomes to recognize the mRNA during the first step of translation. During editing, the introns, the junk inside of our gene, will be cut out, and the exons, the expressed portions of the gene, will be glued together. At the end of capping and editing, we now have a modified mRNA molecule that is ready to leave the nucleus and go to the cytoplasm for translation. The second part of gene expression is translation. During this process, the mRNA molecule, which is a copy of the information from the DNA, will be used to build proteins. Before this process gets started, we have to look at the ingredients needed for translation to occur. The first thing we have to have are free amino acids. Remember, these are the monomers or building blocks of proteins. Without them, we can't build a protein. The second thing we need are ribosomal subunits. Remember from Chapter 3 that ribosomes are the factories where proteins are built. Ribosomes are made of two subunits, the large subunit on top and the small subunit on the bottom. We will also need transfer RNA or tRNA molecules. These molecules carry amino acids at one end and have a piece of code down at the bottom. The last thing we need is our actual mRNA molecule that was made during transcription. tRNAs are one of the key players during translation. This is where the actual translating occurs. These guys are kind of like the secret decoder ring that helps us turn the code of mRNA into an actual amino acid sequence. Each tRNA has a specific amino acid at one end. At the other end, we see an anti-codon. This is a piece of RNA that will line up with a codon, or three-letter word, on the mRNA. Those codons and the anticodons are how we go from mRNA messaging to an actual amino acid. The mRNA is read in three-letter words called codons. Each translates into an amino acid. For example, the codon AUG will translate into the methionine amino acid. It also means start when we look at the mRNA message. Translation is kind of like solving codes. We can use a table to help us actually translate the nucleotide sequences or codons into amino acids. This is the table of codons. We can see that every codon possible is listed in this table. Each codon corresponds to an amino acid. Some codons are repeats. So, for example, this whole block of codons here, UCU, UCC, UCA, UCG, all code for the amino acid serine. We can use this table to help us translate the codons. On one side, we see the first codon position. The second codon position is listed across the top, and the third is on the other side. Translation is a three-step process. The first step is to recognize and initiate protein building. The second step will be to elongate the amino acid sequence, and the third step will be termination, when everything is done and everything falls apart. During step 1, recognize and initiate protein building, our pieces are all going to come together in one giant machine. The ribosome will recognize and assemble around a start sequence, which is always AUG on our mRNA. Once that start codon has been located, the small and large subunits attach to the mRNA molecule. The tRNA that lines up with that codon will also enter the ribosome. The second step of translation is elongate. Remember, we are building an amino acid sequence or a protein during this process. So during elongation, we will be elongating the protein. The first part of elongation is when the codon is read by the ribosome. The mRNA is being threaded through the ribosome, and the ribosome will match a corresponding tRNA to the codon as it moves through. The amino acids are then connected together using a peptide bond, and empty tRNAs are discarded, leaving the ribosome to go be recycled and reloaded in the cytoplasm. Here's a picture of elongation. Again, the mRNA molecule down in the bottom is threaded through the ribosome. There are 3 sites in the ribosome that will be reading the codons and matching tRNA anticodons to that codon. As those are matched up, the amino acids will be connected, making a chain, a small protein, and then everything will shift over. The empty tRNAs will leave, and a new tRNA will be allowed to enter. The last step of translation is termination. The RNA will encounter a stop sequence or stop codon in the mRNA. When that stop codon is reached, translation ends because the ribosome, tRNAs, and protein will all fall off of the mRNA. Everything is released into the cytoplasm. These molecules are all recyclable. The mRNA can be read again, the ribosome reused, and the tRNA is reloaded for reuse as well. Our product, the newly formed protein, is now ready to be transported to wherever it's needed. Let's take a moment to practice the process of translation. Look back at the table that we saw previously. In the table of codons, you should be able to find each of these 3-letter words present in this sequence of mRNA. Translate these codons into an amino acid sequence. Pause the video while you complete this process. We can see that the correct sequence of amino acids is number 2, methionine, alanine, lysine, glutamine, proline. In this video, we covered the 2 steps of DNA to protein. We looked at transcription and translation.